Hey everybody, this is Suzanne Featherling, again, one of your teacher leaders. And today I'm going to go over with you the science behind our first kit for week 1, which is the Photosynthesis Cellular Respiration Lab using algae beads. So like we kind of said before, this lab is really, really versatile. You can use it in a lot of different classes. You can use it in underlevel biology classes. You can use it in AP biology. You can use it in chemistry classes. It's also a great lab for any ecology classes or environmental classes that you might be teaching. So you can use it in a lot of different areas, and we can also beef it up to be pretty intense for some of our higher level kids. And we can also run it in a very simplistic manner. So I'm going to run through just the basics of this lab with you. I have run this lab with my kids in class before and try just give you some tips along the way. So just as a reminder, here are all the labs that you have available to you through this program. So everybody is going to be doing the Photosynthesis Cellular Respiration Lab this week. And also maybe if this is a lab that you'd really like to develop curriculum materials for, you'll have the whole month to do that. But some of these other labs, again, are going to be the ones that you are going to look at and see what else you would like to try. We have the first two that are put up by Mini PCR, the DNA BioBits looking at transcription and translation, and the DNA Glow Lab looking at DNA structure. The next 3 labs labeled here are our simple gel electrophoresis labs with a different theme to each. And then our last 3 are our PCR labs that we're offering, again, each with a different theme. All right, so the photosynthesis lab. The basic premise of this lab is we are going to look at the rates of photosynthesis and cellular respiration under different conditions. So basically, the kids are going to test different abiotic factors. And in the first core lab that the kids start out with, they're really just going to be testing light versus dark. And when they do that, they'll be able to see the balance between photosynthesis and cellular respiration, or if one of the processes is dominating over the other, depending on the conditions that they're in. Then this lab allows the students many different opportunities to test different abiotic factors beyond just light or dark. They can do different colors of light, so testing different wavelengths. They can do different quantities of light, so somewhere in the middle, in between extremely light and really dark. They could do temperature, or they could even possibly do pH. And actually, the pH one is something that I'm going to be trying at home for myself. So I'll talk about that a little bit at the end. But if you're looking at maybe putting this into your actual curriculum, this can easily be related to energy flow throughout ecosystems, or just the interdependence and cycling of materials in between the process of photosynthesis and cellular respiration. And the kids can maybe even go as far as relating this to some of these ideas that are really, really important, such as climate change and ocean acidification, as they start to see the interplay between some of these different raw materials within these two processes. Okay, so one thing that a lot of times you can do if we're in class to get the kids just to understand what's really going on with the CO2 indicator solution that we're going to use is you can give them a cup of the CO2 indicator solution, and the kids can then blow into the CO2 indicator solution with a straw. And when they do that, they're going to see the CO2 indicator solution, which is really a pH indicator, start to change color. And so they can get a baseline of what they're going to start to see and what types of observations they're going to make. Now, this might not be possible because either A, we're not in school, or even if we are, I highly doubt that right now we're going to be able to allow our kids to be putting their mouths on anything, even if it's just a straw. So this could maybe even be something that you demo for your kids. Or even if you're not doing that, you could just talk them through and they could make predictions on what they might think would happen with this CO2 indicator solution. So the CO2 indicator solution that you're going to receive is going to be a reddish color. And what happens is as you blow into the cup, you're going to be releasing CO2, and that CO2 is going to turn into carbonic acid. And carbonic acid is then going to turn this pH indicator solution, which we're calling the CO2 indicator solution, to a yellow color as it turns into a lower pH. What you'll also see in a moment, as it turns more basic, then it's going to turn more of a purple color. So how does this relate to our lab? You are going to get a kit with some algae beads in it. And in our technical video, I'll show you how we're going to prep those algae beads. But basically, you're going to put algae beads in a cuvette here, and you're going to cover them with this CO2 indicator solution. And then you can put them under different conditions. So for example, and I'm sorry that this is a little bit, I'm going to shut off my video camera here. If these algae beads are photosynthesizing quite a bit, they're taking in a lot of light, they're consuming CO2. And if they're consuming CO2, that means we're going to have a decrease in the content of the carbonic acid. If we're decreasing our carbonic acid, our sample is going to turn more basic, and that is going to turn our pH indicator solution, otherwise known as our CO2 indicator solution, to a purple color. If our algae beads are undergoing the process of cellular respiration in a more dominant way than they're doing photosynthesis, for example, in dark conditions, then they're going to be producing CO2. And that CO2 is going to get released into the indicator solution, forming carbonic acid and lowering the pH or becoming more acidic, and our sample is going to start to turn a more yellow color. So that's really all the basic premise is to this entire lab. And what the kids can start to do then is change the abiotic factors that they're testing and see if there's a variation in the change of these samples that they've set up. So one thing you can start to discuss with your students is the balance between the two processes of photosynthesis and cellular respiration. Now, the kids will probably be able to tell you that during the day, these algae are going to be photosynthesizing. And so therefore, that will be the dominant process. Sometimes I feel like the kids kind of don't really think about the fact that plants are also doing cellular respiration, or algae are doing cellular respiration. And that what this will show them is that the process of cellular respiration is kind of always at like a baseline level. And so what happens is if these algae beads are only exposed to the dark, like in nighttime, then you're going to have the cellular respiration be the dominant process, but it's sticking at a baseline. And it's only when these algae beads are exposed to light that you're going to have photosynthesis override even that baseline of cellular respiration. The compensation point is when those two processes are pretty much in equilibrium. And at that point in time, you're going to have samples where the CO2 indicator solution is looking like it's at one of those neutral pH colors, which looks to be a little bit more amber-like. So when we're looking at this, what does it look like with your beads? So you have this red solution with your CO2 indicator solution. That's going to indicate that the beads are more just like at an ambient light, and they're a very neutral pH, and both of these processes are in balance. And this is how you're going to want to start your samples. So if the kids maybe have like 5 cuvettes that they're testing, let's say they're testing different wavelengths of light, you would have 5 cuvettes set up, each with algae beads in them, and you would want this pH solution or the CO2 indicator solution to be at a very neutral pH. Then you could have some of them under light conditions. You're then going to have that consumption of CO2 and that decrease of carbonic acid. And the pH indicator solution, or I'm sorry, the CO2 indicator solution is going to turn purple. And then you're going to have some that are not photosynthesizing at all. So that process of cellular respiration takes over. And this then becomes more acidic as the formation of carbonic acid occurs and the pH turns yellow. Now, if any of us are even lucky enough to get back into the classroom, or maybe if you want to use this in the future when things are back to normal and you have a spectrophotometer and you would like to use that maybe with some of your older kids, what we can do is we can take a baseline absorbance reading with these samples, and then you can take absorbance readings between those other samples that you have as well. And you will see if you just have in your basic core lab light versus dark, the absorbance values of the light cuvette will increase, while the absorbance values of your cellular respiration or your dark cuvette will start to decrease. Now, if we're doing these labs at home, or we don't have access to a spectrophotometer, or you feel like that's just a little bit too much for your class, the kids can use the exact same graph, but they are going to turn this y-axis here into pH instead of absorbance readings. So going back to this slide for a quick second, this pH strip here, it's a little chart that the kids are going to be able to use. You will receive this within the kit. So if you want to take absorbance readings, you can get those from a spectrophotometer. Obviously, right now, I don't think that's feasible. So what the kids are going to do, or what you're going to do, is use this pH indicator chart, and then you can approximate the pH of your samples as they start to change color. So when that happens, and we're looking at this graph here, the kids can still graph this, but they're going to use pH values instead of absorbance readings. So, as I mentioned before, a couple of things. The first part of the photosynthesis cellular respiration lab is a core lab, and it's just the kids. It's very simple. They have 2 cuvettes. One is put under the light, one is put under the dark, but they both start out with that fresh CO2 indicator solution that's at a neutral pH. And then they take measurements every 5 minutes to see how their samples are changing over about a 30-minute to 45-minute period, whatever kind of works for you in your classroom and the time that you'll have allotted to you. But then if you want, the kids can then do an extension lab where maybe they could test one of these 4 questions. So they could look at light intensity. I can show you how I'm going to do that in the technical video that I'll offer. They could do light color. So they could wrap their cuvettes in different cellophane colored lining. They could look at temperature, set up little water baths. The pH one is one that I am going to try, and it could maybe be an ultimate fail, but I'm going to actually soak the beads in vinegar and Windex and then rinse them off and see if their rates of photosynthesis and cellular respiration are going to change. And if we ever really wanted to be ambitious, maybe in the future, you can actually add little snails to your cuvettes to set up a mini ecosystem and see if that is going to change the overall balance of cellular respiration and photosynthesis within the little ecosystem that you set up. Possible curriculum connections that you might want to consider on where to put this. It's a great lab to talk about the conservation of matter and raw materials, or the cycling of energy through an ecosystem. You can talk about the oxygen and carbon cycles if you want to focus on the cycling of matter through an ecosystem. Obviously, at its face, you can use this as your staple photosynthesis or cellular respiration lab. You can look at endosymbiosis and how we have eukaryotic cells that have possibly taken up chloroplasts in mitochondria as their structural organelles within. And then also looking at, therefore, the evolution of how some of these organisms came about. One thing that has always plagued me in AP, there was an old dissolved oxygen lab that was kind of one of the dirty dozen labs in the ecology unit. And this is also a lab that kind of teaches the same concepts. So this is a great lab that I found to substitute in my AP biology class for dissolved oxygen. In my freshman biology class, I'm considering putting this into a storyline of the destruction of coral reefs. So I this lab fits in with the symbiotic relationship of algae and corals, and how algae are really the foundation of keeping these coral reefs ecosystems alive. And without their algae, the corals start to die off. So that might be something that you want to think about. And then also, what's happening with all this climate change and ocean acidification? How might these things actually affect these processes? So I really feel like the reason why this is one of the staple labs that we're using in this program is because it has so many different connections that you can make. Other than that, the only other thing that I want to mention really quick here is the teacher prep. And there will be a separate video that I'm doing that will walk you through the teacher prep and then actually some of the setup that kids might do. So the three main things that you have to do for prep. The first is you have to make the CO2 indicator solution, which really means you have to dilute it. And I will show you how to do that. It's very simple. It will take you about 5 minutes. The activation of the algae beads is what intimidates people a little bit. Basically, you're going to get these beads that are in a light sensitive vial. And again, I will go through this with you in the other video, but you're just going to rinse them with spring water to get them out of the storage solution that they're in. And then you are going to cover them in some of the CO2 indicator solution that you made and let them just sit out for about 6 hours and acclimate to their surroundings because they've been in the fridge. And then you're going to need to put your beads under a light source. Now, because we're working out of our house, a windowsill might possibly work, but you have to make sure that the beads don't absorb heat. It's got to be maybe not direct sunlight then. But if we also will coordinate with some of the ladies dropping off your materials to find a light source that you can put them directly under for about 18 to 24 hours, and also one where they're not absorbing a lot of heat. And then once you're done with that, the beads probably need a little bit of a resting period. And this is, I can't be really specific on because it kind of depends on if you want one process to dominate over the other when the kids start their labs, or when you're actually going to want the beads to really start cycling through both processes. So kind of what I've done in the past is after I've activated the beads, I've set them out at room temperature, just ambient light again for about 5-6 hours. And then I start distributing the beads into cuvettes, which is the third step that I have here. So you spread out the beads into the cuvettes, you put new CO2 indicator solution on them. And then I, in the past, have just let them cycle through day and night by putting them by a window, as long as it hasn't been too hot, until the kids are ready to use them. So sometimes I get all of this done and do number 3, maybe about a week before I'm going to do the lab with the kids, just to make sure that the beads are cycling through as they should. So that means by the end of the school day, they're purple, or the CO2 indicator solution is purple. And then when I come in the morning, it's yellow. But the rest period after you activate them and the cycling within the cuvettes is something that you're going to have to play around with just to get more comfortable with how the beads are cycling. Okay, so other than that, what I would just like you to think about before our meeting on Monday is, what are some things that you feel like you're confident with? Do you have any ideas of how you might want to try this lab? What class might you want to try it in? Does it work into a specific unit, or where do you see it fitting into your curriculum? Also then, what questions do you have? So I'm sure there's something that I'm not thinking of that you have questions on. So what holes have I created for you, or what information have I left out? Is there anything that you would like to go over? And how would you maybe like to start organizing yourself and collaborating with some of the other teachers so you can produce a product for you that's really going to work in the fall for you and your students, or even in the future, whenever that might be when you would like to use this lab. All right, I hope that was helpful. There will be a separate video where I really walk through some of the technical aspects with you of setting up the lab, but that's most of the biology behind this lab. So feel free to check out the manual that you receive in your BioRad kit. I know it's long. It's got a lot of this information in there, but I definitely skimmed over a lot of it. So again, my name is Suzanne. It was great to see you at orientation, and I will see you on Monday. Thanks.